we can open the floor now for general comments or general discussion on any of the material we've been through today. We've covered a lot. So maybe there's certain technical things that weren't clear that you're sort of like, can you just tell us a little bit more about something? Or maybe there's a wider topic or a wider topic that you want to talk about, or just general bioinformatics questions or advice, or anything like that, or motivations. Why do we do it this way and not that way, and so on. Feel free to drop drop questions into the various chat groups. You got a request for Panaru and Bacta.
Bacta is in there, and Bacta is pretty awesome. I read it on 60,000 or so genomes. So I'm excited to report that to Oliver and like let him know, like, hey, Bacta worked great. And Bacta is kind of a bacterial annotation, like a substitute for Proca, whereas Proca like gives you good enough results very quickly. Bacta takes a little bit longer and gives you a slightly better annotations compared to Proca.
Yeah.
Panaru, I've seen that one, and I'll look into it.
Okay. Yeah, I think standing up Bactopia to run, I think it's like, if you gave me a fresh VM, how long would it take me to set up Bactopia and have some data done, like run through 10 genomes or something? 2 hours, I think. If I use Mamba, probably like half the time. But if I use straight up Conda, because it has to sit there and build all the dependencies for all of that stuff, it takes a while. But yeah, it's like an hour or two to get up and running. So that's blank VM, install Bactopia, download all the data sets. It's got a Kraken database in there, all sorts of stuff. And then I'm off and running with my read data, download that on and run through it. And maybe in an hour or two, I can do like 10, 20, whatever. So it's really good to get up and running, and it'll provide you with a lot of basic critical results that you'll need. PGAP. Request for PGAP.
I don't think that one's on Bioconda, and I think that will never be on Bioconda. And due to the large database they include in that Docker container, I forget how big that Docker container is. If NCBI were to separate that to where you could install PGAP separately from the database, like kind of disentangle them and get it on Bioconda totally. But yeah, I think that's one that NCBI is going to have to solve on their end before it would make it to Bactopia. There's no reason why you can't use the assemblies and then manually run PGAP though.
If a program isn't in Bioconda, can I install it on a VM from the web? Yes?
Yeah, I think so.
You can install it?
Yeah, I'm just not a fan of making people manually install things anymore. Like I prefer to like point them to a container or a Conda install just to simplify things. I will say I do use containers more than Conda when I'm like running workflows, just because the random Conda hiccups that happen occasionally, especially if you kind of mess up the base environment.
I've had a few. I've become a little spoiled with Conda and containers, and I ran into a tool that was recently published, had to review it or whatever, and it had instructions on, it was mostly Python, and it was like, yeah, it was all handled through sort of pip half something else, but it was baked into the thing. You had to like build its Conda environment from scratch for it, and then give it, and then do the pip environment as well and run it. So there are like 2-3 steps in it, and it was a pain. I was like, what is this nonsense? I have to do this. Why didn't they just, like, what you can do, why doesn't the developer just, even if they don't want to put it in Bioconda for some reason or the other, why don't they just at least give me a Docker file so I can just build a container of their tool instead of going to the README and doing exactly the same steps I would do to build like the container would do. They've gone and written out the steps. I can just make a Docker file with those steps and just give me the way to build a container. So the container would have instead of me typing it out, the container would be built with the conda environments, the pip in the right place and so on, and it's all good to go. So so I think that touches on the question of like, can I install on a VM from the web? It's like, yes, you can do whatever you want installing it. Will you be able to feed it into Backtopia though, Rob? Is that easy to do? I guess you can give it in the next for config.
A particular Docker container?
Like, let's say there's
Or
something, so most of your tools in Backtopia are running in Bioconda, right? So what
yeah.
if I have something that's not in Bioconda and I just want to run it in Backtopia? Okay, that's fine.
Yeah, so I would say do what you need to do, then run it manually. Just because, yeah, it's one of those where I'm pretty strict about that. It has to be on Bioconda bit to include it in Backtopia, and I think that's just a good practice for the bioinformatics community, just to kind of just because it's so easy to get on Bioconda too. Like there's no rigorous review. You just got to meet a certain threshold, a certain bar, and it's like there's hundreds of people waiting to help you out. So yeah, I would probably say, hey, can we maybe work on getting this on Bioconda first and then consider it? And which has happened occasionally.
Yeah,
But PCAP is a whole different behemoth. Like that's a huge thing.
so in terms of how to install, it's dependent on the tool. The tool will have instructions how to install it. You can just install it like that.
Mhm.
I think that covers that. People are heading off otherwise, which is great. That's fine. You will probably get a feedback form. Andreas put it in the general channel, and I'll just drop it here just to make sure people see it. Yeah, please fill out the feedback form. It's really this is the first time we're running this course and we're running with this content. So we're really interested to see how people found it. It's nice that we can do it virtually so that a lot of people can be involved, but it also kind of sucks that I didn't get to actually meet anyone or even really see anybody today. That would have been really good. But yeah, please fill out the feedback form. If there is interest, I don't know, like we can tweak this a bit, tweak the content a bit, and run something similar in the future, depending. Are there any good practices for organizing things in Conda, like don't put everything in base? So this
I'd
is
say
something,
that's rule number one.
yeah, don't put everything in base. This is an interesting one. Like we've touched on this throughout the day. How do you manage that? I'm curious. Rob, I don't think you actually made a statement on it before. How
To
do
me,
you organize your Conda?
the Conda environments, when you do Conda create, those are completely disposable. So sometimes I'm like, here's all the tools I'm going to use. I'm going to just try and create an environment with all the tools that I'm going to use in this one. Sometimes there will be dependency issues and I may have to toss one out. But yeah, if you're creating new environments, I mean, you're free to do whatever. I think that just don't violate the installing stuff, the don't install stuff in the base environment. And Conda usually works pretty smoothly. But I'm free reign on the Conda environments.
Okay.
There is a question from Laura, I think.
I have a question for the panel discussion in relation to the number of citation limits and publications. And unfortunately, many of the times I have to slash the bioinformatic references. Any suggestions? Oh, well, you get rid of the other ones. Don't get rid of the bioinformatics stuff. That's our bread and butter.
That was a hill I was willing to die on with the Backtopia paper. Like I wanted to make sure that table 1 included every tool that Backtopia used at the time and a link to the citation or a link to the repo. Because otherwise they'd say, put it in supplementary, and then it may or may not get indexed and may not get that count. And yeah, that's a tough one.
Oof. How do you manage it, Andrea or Anna? How do you approach that one?
I don't think I've ever had the issue of having to be selective with my references. I've always managed to get everything in.
I think that there are it's good to have a systematic approach as Backtopia offers, because there is a different chance of a tool to be forgotten in a publication, depending on which step you used it. If you use it to make the figure, probably you used it. If it was in the pre-processing, can slip through. So I think it's a good approach to try to put everything, and maybe because times are evolving and pipelines are becoming complex, also some publisher will be more bioinformatics friendly, maybe.
I think there are different elements to this. Is it to give the credit to the developer, or is it a matter of describing how you did the work? Which are two slightly different things. These days, for some journal, they keep changing the way it behaves, and they do have this sort of some of the journals that have that are more print based and have very strict page limits. They have this sort of, they shove everything that's methods into a sort of supplementary methods. I think CellPress does this like star methods thing, and you sort of have tables and you fill everything out in that. Or I think some of the Nature publishing have they have a print version of the references, and then like an extended online version of the references. They're all trying basically different ways to get around this problem. But ultimately, I think the way we write publications, like print publications, aren't the best way of explaining how did we do the work. There are definitely ways of, so that's where the next flow, like the workflows and the cond environments and whatever, and then taking the environments, taking the tools, the scripts that you've used, putting it together on Zenodo or something, putting it on GitHub, minting a DOI of that, and then putting that in your paper and saying, look, if you want to figure out how I did it exactly, it's all here. Go to this DOI, it's there forever. And that's got everyone, that's got the biggest table ever. You can do whatever you want. So I would just basically call out from the paper and just keep saying like, go here, go here, go here, and just redirect out of the paper. Supplementary is, you have to, but it's a lousy way to do it, I think. Lee.
I've really enjoyed the day. It's been a real treat to learn about containers and workflows. So it's been fantastic. I had a more basic bioinformatics question, not having originally trained in bioinformatics, is around the choice of what you use to do particular tasks. So I was having this conversation with colleagues the other day. For things like RNA-seq, what do you use as your aligner? And everyone seems to have their favorites, and there doesn't seem to be a consensus. And I don't know whether workflows like we've been looking at today will help give a bit more standardization or not. But for one of the things I'm interested in is cancer gene expression data. And they've just harmonized recently, the TCGA, they've reharmonized by running, a new pipeline with the original data to try and kind of harmonize it across all tumors. And I guess my slightly rambling question is how do you make decisions as to what are the best things to use to do tasks like RNA-seq analysis? Be interested to know people's thoughts.
I can have a punt first, if you guys want to think. I haven't done much RNA-seq. Helped a few students just run stuff, but haven't really thought about what they're actually trying to do. Generally speaking, when I have a problem with, I have a suite of tools and I don't know what to run, usually I'll have some sort of control material. So a simulated data set or a real data set or something from a publication, something where I go, this is the ground truth, and whatever I'm doing, I have to kind of get into this ballpark of a result. You know, if it doesn't, or if I get something strange, or if I get something that's suboptimal, then I start thinking, well, I don't think this is quite right. So I play around with that. So basically, yeah, like control material in your experiments would help. Just generically, if I have a generic thing, I have no idea what to pick. I think particularly with the RNA-seq, I know the RNA-seq one for NF-core is there, and I don't know if you've had a look at it, Lee, but that's pretty extensive with the options it has. And those guys at the Crick like really hammer it. So I mean, they've probably got some good tips in there of what does work and what doesn't work, what they've included in that pipeline. I think that's a good place to start, is to unpick that. I'll pass it over to the rest of the panel.
I used to do a lot of RNA-seq, but in the past. So what I can share is sort of an outdated experience. That is, sometimes there are tools, because they are successful pioneers, they are adopted way too long. And sometimes the authors themselves, they try to say, stop using bowtie 2 because it's outdated and we did better. There is a HiSat and there is Salmon Callisto. It depends on what you're, but sometimes they survive and outlast their authors' wishes because they were good, but then they have been improved and rewritten, they are more efficient. And sometimes at the beginning, there were a lot of hacks. So you needed the intermediate file to then find out the splicing junctions, and now there are better tools. So I don't have an answer, but sometimes just the rule of the thumb of seeing what is adopted is not the best way. Also because literature is about things that have been analyzed 2 or 3 years ago sometimes. So it's, if you have a look at what's being published now, it's not the best way. So yes, I would probably ask experts in the field, like the NF-core Slack channel can be a very good start. There are sub-channels for every pipeline. So if you have specific questions for the RNA-seq pipeline, you will find their developers patrolling that channel. And,
yeah, I mean, I chose RNA-seq just because I was speaking to colleagues who essentially, a colleague said that, you know, the 2 collaborators were essentially arguing over whether Salmon or Callisto was the best pseudo aligner to use, and essentially my understanding is they're pretty much similar, you know, very similar. So it was an interesting question. It could have been related to anything, not just RNA-seq, but yeah, as all these tools are coming out and as they're getting packaged into pipelines, and I guess my experience before this of using pipelines was using Galaxy, because it's simple and even I can use it. So you can string all these things together, and people have published in Galaxy the workflows, haven't they? But yeah, you know, there's very much a mix of what people are using, and it just slightly worries me that if you take some, you know, some raw data, RNA-seq data, and you want to do differential gene expression, you could get quite different answers by running it through different pipelines, and that slightly worries me, having been from a wet lab background originally.
Yeah, I think what we've been doing in some other hackathons on other subjects, particularly with things like AMR, we've been asking the question of, you know, out of the suite of possible tools, there's no real way to go and, you know, there's 100 tools out there. You're not going to have a domain expert who's going to unpick how each one of them works. Can we establish a benchmark data set that we can run through everything and compare the performance with and see how each and have this available in a way that as new tools arise, we can run it through, and then you just have a workflow whatever to run all of the tools, much like how Andrea was talking about today, and you just look at those results and say, well, how does this new one fare in terms of these metrics? Like, what do you constitute as a good result or an acceptable result? Yeah, I mean, there's plenty of arguments. I guess whatever gets through, I don't know.
I think on the bacteria side of things, we are kind of fortunate that the dust is kind of settled on some core analyses, but like you see on like the metagenomic side, we're kind of like, well, what do we do? This one does this one does that. I had the fortunate enough to be like, I remember when it was, you ran all the tools and then compared all the results. Now,
well, I mean, I think in the isolate space, like with culture, like it's really been dominated by public health microbiology and clinical microbiology, and that's where they've gone and said, they put a gun to your head and say, well, is it this or that? And then you have to say, is it this or that? And that's how a lot of the tools are. But when you want to start talking about, let's go back to the old school kind of comparative genomics days, and we start talking about population genetics, it's again a wild west where it's just like, you can do whatever you want. You can use this tool, you can use that tool. It's just sort of all over the place. But there is this very specific sliver of the microbial space that is like quite tidy. Yeah. So yeah, I guess that's the fun part, isn't it? And I think that's really the that's what I want to think more about, instead of trying to keep writing code to stitch tools together and dealing with differences in file formats or whatever, or making sure that the thing outputs the right thing in the right place. I want the workflow manager to do it for me, so I can then go and ask the question, like, which of the analysis software is the best? What's giving me the result? What are my assumptions and so on? We get bogged down, and hopefully people have appreciated throughout the discussion today that this is a space you can get really bogged down in, of just like trying to get your data through over the line. The result doesn't even make sense. It's just pushing it through. That's like a mammoth effort. I think Rob and I discussed this on the Microbial podcast for a couple of episodes that came out in January. Like, I think as a doctoral student, I spent like 6 months just writing Python scripts to, run a thing, put it into another thing, run it. The result didn't matter. It was just chaining stuff together. It took forever. That didn't go into a paper. That didn't go into my thesis. It was like effectively a waste of time. There was no output out of it. We don't want to do that anymore. And we definitely want to get into these questions of like, well, no, let's get down to brass tacks. Which tool is appropriate for this analysis? Yeah. Oh, and I've sucked all the air out of the room, and we're almost out of time. So is there any final questions from everybody? I think we can start wrapping up and get out of here by around 6 o'clock. I'll pass it over to, we've got Adrian. Want to chip in with the final question?
First of all, thank you very much for the amazing effort, and I really enjoyed it. I know everyone else did as well. And on the same topic, I'm very interested of what is your point of view on whether we need these benchmarking studies and papers that are coming out on different tools. Are they really necessary? Because in the end, we use their opinions or their data to make a decision, or from your point of view, there are just so many tools that it's impossible to really make a good benchmarking.
You can't cover every tool, but you'll see that there's a spread of the possible tools that are out there. So it's not tractable to assess every single read mapper, because there's like 200 of them. But it is tractable to check out the top 10. You tend to see people say, oh, they say Bowtie, they say BWA, they say Stampy, they say whatever. How many are, like, just read mappers. So you could make something that you take the top bunch, much like the way that like Rob's talking about Backtopia. He's collated the bulk of the tool that he sees in his space that are commonly used and put it in there. You can't put every single tool in Backtopia. The same way you could have a benchmark on the top group of different tools. Yeah, I hope that answers your question.
Yeah,
I'll drop a link to the podcast since a few people asked about it. It's just Rob, me, and Lee Katz banging on about. We talk about Backtopia, and then we talk about a part 2 of it. Actually, talks about how things used to be when we started and having to write horrible scripts just to glue things together. If you're someone who don't remember what that was like, then it's a good trip down memory lane. We talk about old school things like really old like RAS, like really old school genome annotation pipelines from the mid 2000s. Any other final questions? Or not? All the chat's pretty quiet. I think everyone's tired. Must be tired by now. I'm tired.
Related to what the discussion was about the benchmarks, I think that when a benchmark is done, it's not only that you read a paper, you read the abstract and you pick their choice, but usually I'm interested in the method they used so that they can adapt the method they use for my needs. For example, and I really think that what Nabil said is the key of doing bioinformatics. I'm now working on a tool for coverage, and I was comparing it with competitors, and I realized that they all work in quite a different way in the end. So if you don't know what's your question is, you might use the wrong tool. And to test the tools, I created synthetic BAM files with 2 reads, because you need a clear yes or no answer. Is this situation dealt with or not? And so when you make these crazy small data sets, you can make the right test to make a choice, because your data set will be always different from the average, from the E. coli data set that someone we used to benchmark the pipeline of bacterial assemblies. Okay, with E. coli, that's the answer, but maybe you have some needs that will probably drive the results towards different tools, and that's absolutely the reason to copy the methodology more than the results of a benchmark.
Yeah, it comes back to one of the things Lee was saying in terms of coming in from a wet lab space, is I think there is in sort of the dry lab computational space, we don't take enough time to think about benchmarking or controls in effectively controls in our experiment. A benchmark data set is a control, positive control or negative control in our experiment. We don't approach it that way, and I think that is problematic. All right, so everyone's saying thanks and everyone's slowly disappearing. So with that, I think we'll draw today to a close. It's just 1 after 6. So I want to have a big thanks to my co-organizer Anna Price, and to Lisa and Mavis, who've been working in the background and answering everyone's emails and helping everyone along throughout the day. A big thank you to Tang, the cloud god, who has orchestrated everything in the background and kept the machines running for everyone just to have a play with all of the different tools and platforms. And obviously, I want to thank Andrea and Robert for excellent talks during the session, and Anna as well for excellent talks today, and running this sort of thing. I mean, we've covered, I think other people take like a couple of days to go through the same amount of material. It's been massive trying to condense all of this down, and everyone's done really well. It's been quite approachable. All of them, there's not only the material that we presented, there's a lot of additional material that's up floating around that we will collate and put up on the website. You will get an email of the full list, all the talks, all the slides, all the worked examples of source code for you to play around with. So this is going to be a persistent resource that you can come back to for help and guidance in this type of development. Hopefully we haven't scared everyone so much that they just decide they're never going to do this. It is worthwhile to have a go and learn these technologies. It's definitely going to be the way we do genomics in the future. So with that, I'll leave any final words from the rest of the panel.
Congratulations to everyone for surviving so far, and it was a good effort.
Yeah,